Confused about whether a population changed because of random chance or because it actually helped them survive? That distinction, genetic drift versus natural selection, shows up all over the MCAT, and mixing them up can cost you easy points. In this video, we'll break down exactly how each mechanism works, when evolution is driven by randomness versus advantage, and how to spot the difference instantly in passage-based questions. By the end, you'll have a clear, test-ready framework for tackling one of the most high-yield genetics topics. Genetic drift is the random change in allele frequencies due to chance events, especially in small populations. This means that, unlike natural selection, which depends on fitness differences, genetic drift occurs purely by luck. So some alleles become more or less common regardless of whether they are beneficial, harmful, or neutral. The key factor here is population size. Smaller populations are much more vulnerable to drift because each individual represents a larger fraction of the gene pool. A classic example is the bottleneck effect, where a population is drastically reduced by a disaster, such as a fire or disease, and the surviving individuals may not represent the original genetic diversity. Another example is the founder effect, where a small group breaks off to form a new population, carrying only a subset of the original alleles. On the MCAT, genetic drift is often tested by asking whether a change in allele frequency is adaptive or random. If the scenario involves chance events with no link to survival advantage, you should think genetic drift. This concept also connects to Hardy-Weinberg equilibrium, because drift violates the assumption of infinitely large population size. Remember the mnemonic, drift equals due to random individuals, fluctuating traits, to keep in mind that this is a non-selective, chance-driven process. Natural selection is the non-random process by which individuals with advantageous traits have higher reproductive success. Unlike genetic drift, natural selection is driven by environmental pressures that favor certain phenotypes, leading to an increase in beneficial alleles over time. For example, if a population of beetles lives on dark soil, darker-colored beetles may be better camouflaged and less likely to be eaten by predators, so they survive and reproduce more, passing on their alleles. This is directional selection, where one extreme phenotype is favored. Other types include stabilizing selection, which favors intermediate traits, and disruptive selection, which favors both extremes. On the MCAT, natural selection questions often involve linking phenotype to fitness and understanding how environmental changes alter allele frequencies. A key phrase to watch for is differential reproductive success, which signals selection rather than randomness. This topic ties closely to evolution and adaptation, as natural selection is the primary mechanism driving adaptive evolution. A helpful mnemonic is select equals survival leads to evolution by choosing key traits, reminding you that selection is purposeful in the sense that the environment filters traits based on fitness. Genetic drift and natural selection both change allele frequencies but differ in whether the process is random or fitness-driven. This distinction is one of the most heavily tested concepts on the MCAT. Both mechanisms can lead to evolution, defined as a change in allele frequencies over time, but they operate in fundamentally different ways. Genetic drift is random and does not depend on whether an allele is beneficial, while natural selection consistently increases the frequency of alleles that improve survival or reproduction. For example, if a hurricane randomly kills a large portion of a population, the resulting allele frequency changes are due to drift. In contrast, if a drought favors plants with deeper roots, leading to their increased survival, that is natural selection. A useful way to differentiate them is to ask, is there a fitness advantage involved? If yes, think natural selection. If no, think genetic drift. On the MCAT, you may be given scenarios where both processes could occur, but you must identify the dominant mechanism. This concept also relates to evolutionary forces overall, including mutation and gene flow, all of which disrupt Hardy-Weinberg equilibrium. Keeping this comparison clear will help you quickly eliminate wrong answer choices. Genetic drift has a stronger effect in small populations, while natural selection can act in populations of any size. The reason drift is more pronounced in small populations is because random events can drastically shift allele frequencies when there are fewer individuals. For example, if a population has only 10 individuals and 2 happen to carry a rare allele, losing just those 2 can eliminate the allele entirely. In a population of 10,000, losing 2 individuals would barely make a difference. Natural selection, however, depends on relative fitness, not population size, so even in large populations, advantageous traits can spread efficiently. On the MCAT, questions often highlight small population size as a clue for genetic drift, especially when paired with events like isolation or sudden population reduction. This concept also ties into conservation biology, where small, endangered populations are more vulnerable to loss of genetic diversity due to drift. A helpful way to remember this is small equals strong drift, emphasizing that drift's impact scales inversely with population size. Recognizing this pattern can help you quickly interpret experimental setups or real-world scenarios in passage-based questions. The bottleneck effect and founder effect are specific types of genetic drift that reduce genetic variation. Both are high-yield examples on the MCAT because they illustrate how random sampling can reshape a population's genetic makeup. The bottleneck effect occurs when a large population is suddenly reduced in size due to an environmental event, such as a natural disaster, leaving a small, non-representative sample of individuals. This can drastically reduce genetic diversity and increase the frequency of certain alleles purely by chance. The founder effect occurs when a small group of individuals establishes a new population, carrying only a subset of the original gene pool. This is commonly seen in isolated populations, such as island species or human populations that migrate and settle in new areas. On the MCAT, you may be asked to identify these scenarios or predict their consequences, such as increased homozygosity or higher risk of genetic disorders. These effects also connect to concepts like inbreeding and loss of heterozygosity. A quick mnemonic is bottle equals break, founder equals form, where bottleneck breaks a population down and founder forms a new one, both resulting in reduced variation and increased influence of chance.